A deep neural network improves endoscopic detection of early gastric cancer without blind spots. Introduction. Gastric cancer is the third most lethal and the fifth most common malignancy from a global perspective. It is estimated that about 1 million new gastric cancer cases were diagnosed and about 700,000 people died of gastric cancer in 2012, which represents up to 10% of the cancer-related deaths worldwide. The 5-year survival rate for gastric cancer is 5% to 25% in its advanced stages, but reaches 90% in the early stages. Early detection is therefore a key strategy to improve patient survival. In recent decades, endoscopic technology has seen remarkable advances, and endoscopy has been widely used as a screening test for early gastric cancer, EGC. In one series, 7.2% of patients with gastric cancer had, however, been misdiagnosed at an endoscopy performed within the previous year, and 73% of these cases arose from endoscopist errors. The performance quality of EGD varies significantly because of cognitive and technical factors. In the cognitive domain, EGC lesions are difficult to recognize because the mucosa often shows only subtle changes, which require endoscopists to be well-trained and armed with a thorough knowledge. In addition, endoscopists could be affected by their subjective state during endoscopy, which restricts the detection of EGC to a large extent. In the technical domain, guidelines to map the entire stomach exist but are often not well followed, especially in developing countries. Therefore, it is important to develop a feasible and reliable method to alert endoscopists to possible EGC lesions and blind spots. A potential solution to mitigate the skill variations is to apply artificial intelligence, AI, to EGD examinations. The past decades have seen an explosion of interest in the application of AI in medicine. More recently, a method of AI known as a deep convolutional neural network, DCNN, a method transforming the representation at one level into a more abstract level to make predictions, has opened the door to elaborate image analysis. Recent studies have successfully used DCNNs in the field of endoscopy. Chen et al. Achieved accurate classification of diminutive colorectal polyps based on colonoscopy images, and Byrne et al. Achieved real-time differentiation of adenomatous and hyperplastic diminutive colorectal polyps using colonoscopy videos. However, in real-time EGC detection alone with blind spot monitoring, the application of DCNN has not yet been researched. In this work, a novel system using DCNN to analyze EGD images of EGC and gastric locations was developed. Furthermore, the authors exploited an activation map to proactively track suspicious cancerous regions and built a grid model for the stomach to indicate the existence of blind spots on unprocessed EGD videos. Background: gastric cancer is the third most lethal malignancy worldwide. A novel deep convolution neural network, DCNN, to perform visual tasks has been recently developed. The aim of this study was to build a system using the DCNN to detect early gastric cancer, EGC, without blind spots during esophageal-gastroduodenoscopy, EGD. Methods: 3,170 gastric cancer and 5,981 benign images were collected to train the DCNN to detect EGC. A total of 24,549 images from different parts of the stomach were collected to train the DCNN to monitor blind spots. Class activation maps were developed to automatically cover suspicious cancerous regions. A grid model for the stomach was used to indicate the existence of blind spots in unprocessed EGD videos. Results: the DCNN identified EGC from non-malignancy with an accuracy of 92.5%, a sensitivity of 94.0%, a specificity of 91.0%, a positive predicted value of 91.3%, and a negative predictive value of 93.8%, outperforming all levels of endoscopists. In the task of classifying gastric locations into 10 or 26 parts, the DCNN achieved an accuracy of 90% or 65.9%, on a par with the performance of experts. In real-time unprocessed EGD videos, the DCNN achieved automated performance for detecting EGC and monitoring blind spots. Conclusion: the authors developed a system based on a DCNN to accurately detect EGC and recognize gastric locations better than endoscopists, and proactively track suspicious cancerous lesions and monitor blind spots during EGD.